Hello, my name is Audrey Lawrence. Thank you guys so much for being here today, for your time and attention. I'm so honored to have been named a Falvey Scholar, and I'm really excited to share my research project with you guys today. So I will be explaining a little bit of my study, which is concerning the use of an orally delivered oxidized cholesterol to relieve inflammatory bowel disease symptoms in a mouse model. And I work in Dr. Bamezai's lab in the biology department. So just a little bit of background on inflammatory bowel disease, or IBD. IBD is an autoimmune disease. So like any autoimmune disease, it's a well-intentioned pro-inflammatory immune response. It's a dysregulated adaptive immune response to non-pathogenic tissue. And so what happens is the body sees something, takes it as a threat, launches a pro-inflammatory response when it doesn't need to. And then this pro-inflammatory response is unable to be reeled back in. That inflammation is unable to be checked, and you reach chronic inflammation, and that's when you really see exacerbation of disease symptoms. So there are two main camps of IBD, although not every patient falls perfectly into one of these categories, but Crohn's and ulcerative colitis, which present differently as shown in this figure. IBD affects over 10 million people worldwide, and there's no cure for it. While there are treatment options available, they usually have either low initial uptake efficacy or a low long-term effectiveness. So if they initially work for a patient, they don't always continue to be really effective. So this is the patient population that really deserves a lot of research and attention to this area. So just a little bit of background on the approach to the treatment that I will be talking about. So lipid rafts are a microdomain of the plasma membrane, and they're a dynamic domain in the membrane, and they're made up of a lot of fatty acids. So they're very rich in cholesterol, transmembrane protein, etc. And they are really important for immune cell communication and immune cell signaling. So when you want to launch a pro-inflammatory response, you need to activate those T cells, tell those T cells, those pro-inflammatory T cells, what they're doing and how to do it. And for this communication event to take place, they need to make an immunological synapse. And at this synapse, this lipid raft is really important in directing all the molecules where they need to go. So because the lipid rafts are so important for this T helper cell communication, so for that inflammatory response, the idea is that if we can alter the structure of this lipid raft, alter the function of it, we might be able to interrupt that signaling and diminish that inflammatory response. So you see here we have cholesterols. So my project is dealing with an oxidized cholesterol. So an oxidized cholesterol is a bulkier cholesterol. So I work with 7-keto cholesterol, so it's a little bulkier at the 7th carbon. And the idea is that if I can displace it where that regular cholesterol is, I'll be able to disrupt the membrane order and affect that T cell signaling. And this was a question that was previously asked in vitro in the Bamezai lab before I came to Villanova. And it was actually proven and published that in vitro 7-KC, that bulky cholesterol, is able to downregulate T cell clonal expansion. And so my project is really going to ask that in vivo. And this is some data from that study showing the decrease in T cell proliferation. So this is Dan Schaefer in the Bamezai lab that was published before my project. So I'm looking in an in vivo model. So I work in a mouse model of inflammatory bowel disease. So these are interleukin-10 knockouts, and interleukin-10 is a potent anti-inflammatory signaling molecule. So when they don't make any IL-10, so that's the knockout, they have a lot of unchecked inflammation because IL-10 is one of those homeostasis, reel it back in, wanna kind of players. So IL-10 knockout mice get severe IBD. So that makes a great disease control. And then C57 mice are just regular healthy mice. And so I'm working with an oral dose of 7-KC complex. So my treatment group is going to receive 7-KC orally, and my control groups are going to receive saline orally on the same treatment schedule, everything there. What I'm expecting to see after the study completion and when I'm doing my analyses on their gut tissue microbiome composition is the disease control should have pretty severe IBD symptoms. So that's tissue histology markers, a disrupted microbiome, an injured microbiome, and inflammatory markers of their cytokine profile. So that's their signaling molecules of their T cells. C57, the healthy controls, I'm really expecting no IBD symptoms, a healthy microbiome, and low inflammatory markers. And then my treatment group, what we're waiting to see here in this presentation, is an amelioration of symptoms, both in the histology, but also in that microbiome. Can it recover all of that injury, or some of that injury at least? And looking to see decreased inflammatory markers, at least from the disease control levels. So what I started with was hematoxylin and eosine staining. So this is just general staining, just to look at the tissue a little bit clearer under a microscope. And so I'm going to start over here on the right. So this is the healthy gut tissue, and this is large intestine tissue from those mice that I was just talking about. And this is in the middle here, we have our disease controls, and then on the left over here, we have our treatment group mice. So on the right, you can see there's ordered lumen structure. So this is all, ordered and regular. The epithelial lining is relatively thin, as opposed to this middle guy, which is our disease control. You can see really thick epithelial lining, a lot of disorder happening here. And then this is our treatment group. You see a little bit of a restoration of that lumen space, a little bit of a restoration of that thin epithelial lining, which was really exciting. And like I said, this was the first kind of test that we did, look-see into what I was doing. So this was really, really exciting. And then this is just a quantification of that image. So you see the treatment group kind of pulls down on that thickness, that inflamed tissue. So that's exciting as compared to the healthy control versus the disease. So this is just like a big picture image showing you a heat map of the microbiome. So like I said, I was also interested in the microbiome composition and how 7KC is able to affect that. We all know how important the microbiome is. And here is just showing by abundance score and a heat map the ability of 7KC to differentiate these two from each other. So these two are the same genotype. Our disease and treatment group, you remember, are the same genotype. But they are statistically or significantly different by their abundance score. And same with the healthy control. But the treatment group and the healthy control are not significantly different from each other by abundance score, which is very exciting for us. And then this is just a little bit of a zoomed in lens on some of the specific strains that we saw that difference in. So you can see here our treatment group are these gray bars, healthy is orange, disease is blue. You see a definite change, a shift between the disease and the treatment group. And the only difference there, again, is that 7KC, that oxidized cholesterol. So you do see kind of a switch and a little bit of a recovery of the microbiome, which is definitely exciting. Specifically here, I just wanted to highlight quickly, this EFACalis is a known colitis causing bacteria. And as is C. diff, and there's not really much to report on here, but I just thought I'd show that. But you do see a restoration, a big difference down into the treatment group. It's a much lower level than in our disease control. Looks a lot more like the healthy controls. So that was definitely exciting. This is a heavy hitter, bad bacteria. So we were excited to see that. And then finally, I did some immunohistochemical staining. So this is really exciting to do. So I have the 3 groups here, and each box is its own stain, and then this D box is kind of the overlaying of all of them. So I'll start over here. So the blue staining is just a generalized staining the nuclei, showing us where the tissue is and where the staining could potentially be. This green is IL-17. That is a pro-inflammatory T helper cell marker. So that's kind of showing us what are our pro-inflammatory T cells doing. And then down here, the red is a FOXP3 fluorescently labeled antibody stain. So that's going to show us what our Tregs are doing. So this homeostasis, reel it back in T cells. So as you can see here, healthy on the right, disease in the middle, and treatment on the left. You see a lot more of the T helper cell expression in the treatment than you do in the disease, In the disease, you see a lot more inflammation and pro-inflammatory signaling than you do in the treatment and the healthy, who kind of look like each other. So just to wrap this up quickly, I know this was just a little glimpse into, a 3.5-year project, but this is a really, really exciting direction that we're going in. And I'm really excited about this project. It has a lot of potential in the future, looking at, maybe a potential drug trial. Obviously, we're very preclinical right now, but the results are definitely really exciting. And I'm working on getting some more data right now, actually. So science never stops. So really excited about this. And this is that 7KC again. I have so many people to quickly thank. First of all, thank you so much to Falvey Memorial Library. I knew nothing about immunology when I got here, and I really owe so much of my literature dives and my ability to just really be uninhibited in the questions I could ask and the things that I was able to learn from different scientific journals, etc. Endlessly grateful for that. Dr. Bamezai, an incredible research mentor. Everyone at the Center for Undergraduate Research and Fellowships has been so amazing. So thank you so much. And if there are questions or time for that, I would love to field those now. Thank you guys so much.
So thank you so much, because one of the things that really came across in your presentation is how impactful your research that you've done in the last 3.5 years could be. So I appreciate your enthusiasm. So I'm curious, what were the greatest challenges for you in doing this research project?
That's a great question. I would say most recently, a remote world has been very challenging. So I just wrapped up another in vivo trial, another mouse trial that in an ideal world I would have completed in the summer with a VERF. So the timeline is a little bit challenging, but I think in a way it was almost a bit of a blessing as well, because it really allowed me to try to piece out every bit of data I could from that first in vivo trial. So I think that in a lot of ways it really grew, and my logic and cognitive pathways going into the second trial were a lot better formed and a lot more intentional. So I think in that way it was a good thing, and I definitely am excited to continue to try to work through this new trial. But definitely the pandemic was a little scary for a while in terms of having mice and that whole approach, but it's been good in the end, so I'm grateful.
So, just thinking about maintaining the mouse colony during the pandemic and your knockouts, were there any unfortunate, unforeseen problems because of that?
So we don't have a running breeding colony of our own 10 knockouts. So that wasn't as much of an issue, but definitely planning with Jackson Labs is where we get our mice from, and planning the delivery was a little bit of hiccups there as well. So overall, it definitely could have been worse. I'm glad that we were able to, respect all their little mouse souls. And I was really nervous that we were going to get shut down when I had mice. So I was a little bit cautious about when we'd order them, because, we definitely want to make all of our projects really ethical and meaningful. But so that was an issue that we didn't run into, which I'm very grateful for.
So what are your future plans for your research?
Yeah, so I'm still wrapping up. We just finished another in vivo trial, like I said. The plan is to hopefully publish sometime soon, finish that up in the summer. And then I've completely fallen in love with immunology and autoimmune research. So my plan long-term for my life is to do an MD PhD and hopefully go into like IBD research in the future.
That's very exciting. Congratulations. So there was one other question about you felt uninhibited in dealing with your questions that you could ask at the library. Could you say anything else about that?
Yeah, for sure. It was honestly a very fun process. So I mentioned I didn't know a lot about immunology when I first got to Nova. I think immunology is one of those beautiful fields that you know nothing until you know everything. It's really hard to have a surface level understanding of immunology. You really have to like dig deep to really understand it. And so definitely, starting as a freshman, even in the remote pandemic VERF, where it was basically all literature searches, being able to ask any question I could think of and knowing that I would have access to really any article, really any journal was very exciting, honestly. And I feel like I would find the most like niche like journals and be like, there's no way I'll have access, and then like go and look and either have access or be able to get it through the interlibrary loan. So that was very fun, and I definitely felt like anything that I could think of that I wanted to learn or anything that I rabbit holed myself into, I had access to, which I was never was lost on me how blessed I was for that. So thank you guys so much for that.
You're very welcome. So thank you very much, Audrey, for your presentation. It was really interesting.